So in this presentation, I'm going to be describing some real-world molecular workflows, for example, to calculate binding and formation energies. This is actually going to be the first of 3 talks we're covering today, and they're going to steadily increase in sophistication and complexity. So after this, there'll be one on bulk and monolayer germanium selenide, and then one on a sophisticated workflow for general 2D materials. The goal of this is to inspire people to move from the simpler tutorial calculations to real research calculations. And actually, in many cases, this is a very small step. So this is what I aim to show in this talk today. And that's really my goal for this short presentation, just to illustrate a research level workflow for molecular systems. So isolated systems for the most part, where we're going to be using Gaussian basis sets and wave functions obtained from PySCF in this particular workflow, although of course one can use other codes for that as well. Just to get started, I want to show 3 examples from the literature. There's actually many papers out there now running some variant of the workflow that I'm going to be describing. And I have picked these 3 examples because in addition to doing the benchmarking and analysis and science that's in the papers, the authors have done a very good job including the details that are needed to reproduce their calculations. So things like the number of walkers that were used, what was the time step, details of the basis set, and so on. And I've also picked this from a variety of different codes from different groups so that you can see that there is a commonality to the recipes that are being used, and actually they are actually very accurate. So one thing you could do is look at one of these papers and either reproduce part of them using a QMCPACK, which I would hope is at least as efficient as the different codes here, and certainly with the workflows should be a little bit more straightforward, more convenient to run. Or move to the next step and do a related system where perhaps quantum Monte Carlo has not been applied, and then also importantly where other methods have difficulty and we would like some more insight. So just to show some examples, first of all, this study is actually up on the Chem Archive, so it's not published yet. It's presumably under review at a big journal. And this is a study of ligand pocket interactions. So essentially in the model calculations that are done, small molecules binding to larger organics, and you can see some in the figure here. And in addition to the analysis that's done in the paper on the sort of more applied science side, there's one striking result that comes out on the benchmarking side, which is a very good agreement between both quantum Monte Carlo and one of the local coupled cluster variants. So they're proposing using both of these in combination, as they have slightly different approximations, well, very different approximations and sources of error. One of the surprising things, though, is how close these come with the standard recipe of wave functions. So what's shown here is the interaction energy, the binding energy, kcal per mole, for these sort of more than a dozen systems that were studied. And their quantum chemical results are in black here, and there's some estimate of the uncertainty on them. And then the diffusion Monte Carlo results at a particular time step in this plot are in blue, and that's a real statistical error bar, if I remember correctly. These are in close agreement, apart from a couple, and obviously there's analysis in the paper done around that as to what the sources of difference could be. When we look at the quantum Monte Carlo, what's done though is actually something that we've been describing. It's a single determinant of LDA orbitals. They used our correlation consistent effective core potentials, which we heard about a few sessions ago, and they go down to 0.015 AU as the smallest time step in the paper, which is very consistent, which is what we had in our tutorials. Moving on to the second, actually older set, this is a study of the S22 test set, which for anyone who's not familiar, it's a benchmark set for non-covalent interactions. So the test set includes hydrogen bonding, dispersion, bonding of van der Waals, and some in-between systems. And this was done actually back in 2008. So it'd be very straightforward to run, some or all of the molecules in this test set today on fairly modest resources. And even then, very accurate results were found. There they used Hartree-Fock orbitals and an older set of effective core potentials. This paper also has some studies of a number of other systems in it, so I'd recommend reading if you're interested in weak binding between molecules. That's obviously a challenging area and one needing more research. And then finally here, I've cheated a little bit. This is not a study of isolated molecules. This is the study of the lattice energies of molecular crystals I pointed out in my Introduction to Quantum Monte Carlo talk in the second session, where a test set has been done for the X23 test set has been computed with diffusion quantum Monte Carlo using the standard recipe. And the authors claim that this is providing the same or better reliability as experiments. So that's quite a strong statement. And in a physical review letters, and you'll see that the recipe there is really very, very similar to what we've been describing both in the solid state tutorial and our molecular tutorials. So these are three examples where the standard recipe that we've been describing provides a very good starting point for calculations. And that's what we're now going to use in this example application. So this is actually a study that's still ongoing. And while we're focusing on one part of it, I should just point out there's a broader investigation here where I've been asked to study a larger set of molecules and different geometries and essentially find a reliable modeling approach that preferably is cheaper than doing a pure quantum Monte Carlo. So what I'll note here is, of course, I spent a lot of time describing some of the motivations for looking at this system. So this system is the adenine dimer, which is, of course, has a very important experimental literature, but also quite a significant history in the quantum chemical literature. So, for example, that paper with the S22 set has some related quantum Monte Carlo calculations done. And there's some known sensitivities in this system. For example, does the dimer stack through van der Waals, or does it hydrogen bond, or do something in between? This is a sort of well-known sensitivity. The reason for focusing on this as well is that it's a 30-atom system for the dimer and it has 100 valence electrons. So the calculations are obviously more expensive than one might run in the virtual machine, but they're not very expensive overall. I set these up to use 64 cores for less than 24 hours for each of the individual runs, because that fit very easily on one node on a CPU system we have here. And hopefully many of you have got access to resources like that. So this is very straightforward. It could probably be optimized further, but I set it up to very quickly get some diffusion Monte Carlo results that I'm going to be showing you. So what's our strategy here? And this is really generally applicable. So the idea is we're going to compare some diffusion Monte Carlo results with those of cheaper DFT approximations, other less costly many-body methods, and quantum chemical methods, and to do so for a variety of different geometries and compositions eventually. And then try to keep our fingers crossed that there's one that matches well. Obviously, one can also think about machine learning and other approaches. So what we're doing in the study I'm presenting here is to start with relaxed geometries that have come from different DFT approximations. And then we're going to use those to compute the energies with the various electronic structure methods. And then ideally, we'll find a more approximate method, a cheaper method that tracks the diffusion Monte Carlo results very closely. And so then that's validating that method. Of course, there are many other routes to doing this. I showed an example where people had taken molecular dynamics snapshots in the past. One can obviously put more effort in here, the more important it is to know how closely these different methods match. Something else that was happening in the background here is generating trial wave functions with different DFT approximations so that in the eventual study, I'll be able to comment on how sensitive the results are to these different choices. And that's exploiting the variational properties of quantum Monte Carlo which most other methods, of course, don't have to rely upon, where we can actually see how the choices that we're making in setting up the method are affecting the accuracy. So that's the strategy. Of course, it's generally applicable to other molecular and solid state systems. How do we go about running this? Well, the good news is that we really only need to make some simple generalizations to the molecular tutorial. Just some very simple, basic Python improvements is already enough to do what we need. Of course, one can start with the workflow that I've included in the repository, but I'd also recommend, the first time you do this, build up the workflow step by step. Make sure you understand every step you're putting in there and understand how well it's working in the Monte Carlo. So I'm now going to talk through some parts of the workflow script that are different from those in the tutorial. And these are actually all small differences. You see here the example files are in session 6 molecules. And the first part I want to talk about is actually the settings at the top of the file. So this is where we set things like the directories, where I want to find pseudopotentials, how often Nexus will poll for results. I usually turn this down so that we're not polling the queuing system very frequently on the shared computer I'm going to be running this on. Administrators don't always like that. And then the key difference I want to talk about here is that instead of running on a machine called WS and a number, such as WS4, where there's assumed to be no queuing system, and Nexus will simply run calculations locally, tetrising the calculations in with the core count you give it automatically. I'm specifying a different machine here, and baseline is a CPU cluster running Slurm, and there's settings elsewhere in Nexus in the machines.py file, which tell it that this is a machine with Slurm, and you need to submit jobs, and that's how it is then going to manage the calculations. And I'll note here, there's also a way of routing in like project and account codes into the chopper job files if you need that to run on a particular machine. Something else I would highlight here that's useful when you're running on a shared group or maybe a university resource is you can limit the number of submitted jobs that are submitted simultaneously. So in this case, I've set it to 24, which is the limit one can have in one of the queues on this machine. And of course, on most machines, there's both the limit that comes from how the administrators have set up a machine, but sometimes also there's some social contracts where other users prefer you don't have more than 10 jobs in the system. So that's one way of handling this. And we're going to be doing a parametric study, so we can potentially generate a large number of jobs here. So this is actually quite an important setting. So what do we need to do now to scan over structures and functionals? Well, essentially all we need to do is add some loops around the example workflow we already had in the molecular tutorial. And that's what's going on here. So I've actually got a set of functionals that I'm going to run. I've sort of built this up over time, and it's grown even more since I took this screenshot. And then I have a subset that I'm going to do the QMC runs on. Then we have this very simple for loop looking for all the files in a directory called structures. And I'm going to have a set of X, Y, Z files in there for the molecular structures. And then once we found them, I've got this loop over functionals, and then as I've commented this out, so the syntax highlighting didn't panic too much. Here is where we would then run PySCF for each individual structure. And then somewhere further down, obviously I've commented this out for the PowerPoint, so it's not too long. We then have a simple filter, and for the subset, we then start to run the conversion steps and the quantum Monte Carlo workflow that I'm about to describe. So what we've really done is add a few loops around the sample workflow we had in the tutorial. So what then about the quantum Monte Carlo workflow? Well, all that's essentially changed from the initial tutorials that we had for small molecules was that I have increased the statistics, and now I've been very careful with the different stages of Monte Carlo that are done so that I can verify that they are working well. So as was described by Jaron Krogel in describing Nexus, we can use that Nexus to set up a series of interdependent calculations of recording any number of codes. And what we've done here on the QMC workflow side is essentially to gradually build up complexity in the calculation that's being run, actually very cautiously. Since this was the first time I had run these kinds of molecules in quite a while, I wanted to make sure that I'd easily be able to see that progress was being made. So what steps are in the workflow? Well, actually, I have a very paranoid determinant-only variational Monte Carlo run, so no Jastrow function. And I use that just to make sure that the output from PySCF was being properly handled, there were no issues, and to start getting initial timings. And then we have a series of steps where I did 1 and 2 body Jastrow optimization. I then did a separate VMC check run. I did some further wave function optimization just to see if a few more cycles would get anything more from the system, or perhaps additional samples would help the optimization converge better. Finally, in terms of optimization, we added a 3-body Jastrow and then repeated the same thing. And then eventually I did diffusion Monte Carlo for 3 different time steps. I'd like to highlight here is that by using the workflow system and the dependencies set up in the workflow, this allows for simultaneous execution of some of these steps if your machine is wide enough and the queuing system allows it. So just to talk through that here, I've got this initial calculation, which takes the orbitals from PySCF, and you see the Jastrow block is empty here, but it only depends on the orbitals. And then here we have an optimization block, and again, which is a different set of inputs, setting up the Jastrow function. And again, same dependencies. So these 2 calculations will be submitted as jobs almost certainly at the same time, and potentially they'll even run at the same time. So even though I've got all these extra check runs in this workflow, it doesn't actually hurt the throughput very much at all. Just to talk about the settings I've put here, these are extremely cautious. I used 1,000 equilibration steps. I left 100 blocks so the block-based statistical analysis will be robust. And then I put a formula in here for the number of steps that will be taken by each walker so that independent of the size of the machine that I'm running on, we'll always get the same statistics. Then down here, we have adding the 1 and 2 body Jastrow function, actually just like in the tutorial, and a very healthy number of samples to start out with. This is most definitely overkill for this system, but as I said, it's being very cautious, and through this approach, actually everything worked first time. How did I build and debug this workflow? For anyone who's used to doing sort of development, I think this would be very straightforward. Basically, I did it step by step. So starting with an existing workflow, I just commented out all the parts that I didn't want to run initially. So I took one initial geometry file. I then made sure the PySCF calculations were okay. They were. Then I added steadily more steps from the overall QMC workflow before eventually saying, okay, I'll go for it and run all the diffusion Monte Carlo. And I'll note that, I ran 2 fairly conservative time steps here. This first one is actually smaller than what was run in some of the examples that I showed from published papers earlier. But I also wanted to show study a much larger time step, because for the same overall compute cost, this will give much better error bars because of the shorter autocorrelation period. And of course, I care about the differences in properties of these systems. I'm not only interested in the energetics of them. And so just to note again, when rerunning the workflow, Nexus is going to look to see which calculations have been completed and will only run the new calculations. It doesn't have smarts to see if you've changed a parameter in one of the calculations, and so you handle that just by removing those directories, and then it will rerun them. And of course, once I had done this for one geometry, I put in basically a dozen and then ran the whole workflow, came back after a weekend, and then set about doing the data analysis. So let's touch on a little bit of sort of science results here. So I mentioned in the introduction that the geometries strongly depend on the functional. And what I'm showing on this in this plot on this x-axis are the density functional approximations that have been used to generate the geometries used for the results in that spot. And those to the left of this gray line are actually snapped geometries, and those to the right are hydrogen bonded. And obviously, one can keep adding different DFT approximations here. So apologies if I haven't included your favorite in this plot. And then what I'm showing here on the vertical axis is the energy. So lower is more strongly bound to relative to the B3 lip geometry result, which I picked arbitrarily. And I'm showing here a subset of results. I'm just showing the results for 3 density functional approximations and one of the diffusion Monte Carlo results at one time step. And there's already a lot of data, and that's the point we'll come back to. So the diffusion Monte Carlo results are these red bars with the red circles with the arrow bars. So just a little bit bigger than the size of the symbols here. And of course, if a density functional approximation was closely tracking this, its points would follow the same trends. And we see that PBE0D4 does in this case, but B3-lip and PBE are much further off. So one might be tempted to conclude that much of the difference here is due to van der Waals and dispersion handling, which of course is very reasonable given what we know about stacking in the system, but obviously that's not the entire story. It definitely helps the DFTs. That shouldn't be a surprise. What I want to note is that this is just a small subset of the data that I generated in just one weekend's worth of running. So running these workflows and doing a parametric scan, you can produce not just a substantial number of submitted jobs, but a lot of output data. So if you're not used to these kinds of parametric surveys and calculations, my advice would be to make a plan at the beginning, think through how you are going to analyze the results statistically, and then how you might plot them eventually, because you can quickly get overwhelmed with data. So think about computing means, mean absolute deviations, and so on. But already with this example, with not very expensive calculations, we can already see that one particular density functional is doing better than a couple of other ones. So that's all I actually wanted to include for the isolated molecular examples, but I did want to touch on the molecular crystal result, because if one has done the isolated molecular case, you might be wondering, well, how large a step is it to now do the periodic solid state case, particularly for molecular crystals? And the answer is that actually you need to do very few changes to the workflow. It's really very similar. And if you're interested in this, there's a very good description in the supplemental of the paper of what was done. And what one would do here is essentially switch to using periodic boundary conditions and quantum espresso, as was covered in the last session. And then you see here the recipe that was used in this paper is essentially what we've been using for the molecular case. The one difference is the finite size correction scheme that was described in the solid state session. And of the range of different ways of doing finite size correction, for this paper, the authors found that actually one of the simplest approaches worked very well, one of the older approaches. So that's really straightforward to apply. And also, they indicate actually the overall costs of running these calculations were, I would say, very reasonable. So the message here is that this sort of standard recipe is actually giving very accurate results, and certainly an excellent starting point for these calculations. So just to summarize, I hope I've made clear that the tutorial workflows we presented for the molecules are actually very close to what you need to use for research level calculations. When one starts moving in this direction, I'd really recommend building up the workflows step by step. That makes it a lot easier to understand what's happening and debug in case of any issues. And then, finally, just to point out that this standard recipe of using single determinant trial wave functions and diffusion Monte Carlo is a really excellent starting point, and we'd advise everyone to start there before looking at more sophisticated wave functions, because not just ourselves, but many others in the community have found this to be a really excellent an insightful starting point for a broad range of molecular and also solid state systems.